Here in Kobe, where a municipal program to foster medical industries is being pursued, a large facility is under construction in the Harbor District. The RIKEN Advanced Institute for Computational Science will house what aims to be the fastest supercomputer in the world. It has been nicknamed K, the sound of the Japanese word for 10 quadrillion. The advanced supercomputer will be capable of performing 10 quadrillion operations per second, an ultra-high speed that is referred to as 10 petaflops. Using this supercomputer, scientists will be able to perform precise simulations that will begin to unravel the essential mysteries of life. A heart reproduced as a precise replica of the actual organ. Red blood cells and platelets flowing through a blood vessel. The complex interwoven neurons within the brain. This simulation faithfully reproduces the transmission of electrical signals. In the future, simulations like these will be performed for all the cells and organs of the body, with the goal of reproducing the functions of life and bringing progress to the fields of medicine and life science. It represents advanced research at levels unmatched anywhere in the world. We will explore the potential of this grand project and the impact it may have on our lives. RIKEN is a public research institution with over 2,700 scientists performing everything from basic to applied research. It is Japan's only comprehensive natural science research institute. Since 2006, it has been engaged in a unique research effort. The Next Generation Integrated Simulation of Living Matter R&D project. A coordinated effort involving 14 universities and institutes. The project involves 300 specialists from a wide variety of fields, including mathematics, biology, fluid mechanics, computer engineering, and medicine. Using the new supercomputer, which will be fully operational in 2012, the project will perform simulations over a range of scales, from human organs and cells down to the proteins that are contained within them. The project is comprised of 6 research teams: the molecular scale, cell scale, organ and body scale, data analysis fusion, brain and neural systems, and high-performance computing teams. Elite scientists in each of these fields will boldly pursue the mysteries of life. Until now, research on the essence of life activity has been pursued through the minute examination of the human body. The body is comprised of 60 trillion cells. Within the cells are long strands of DNA, just 2 nanometers, or 2 1/millionths of a millimeter in width. This DNA is thought to hold the key to the functioning of human life. However, in recent years, we have come to understand that the structure and dynamics of proteins created on the basis of DNA sequences are deeply connected to life activity. The 1997 Nobel Prize in Chemistry was symbolic of growing attention to the importance of the dynamics of proteins. Paul Boyer and John Walker shared the prize. They had focused on a protein called the ATP synthase. This protein spins like a precision machine within the cell to perform an essential function. The ATP synthase lies in the part of cells called mitochondria. Here it creates ATP, the source of life's energy, and performs a role indispensable to life. However, since proteins are exceedingly small, they cannot be seen directly. A technique known as X-ray crystallographic structural analysis is used. X-rays are directed at the protein, and its structure is deduced. Using this technique, the structure of the ATP synthase was obtained. It looks like a jellyfish, about 20 nanometers or 2/100,000ths of a millimeter in length. From this shape, it was concluded that the ATP synthase spins with the circled portion as its axle. In order to make the spinning visible, a marker 100 times the size of the protein was attached to the axle. This is how it appeared. In this fashion, it was confirmed that the ATP synthase actually spins. The ATP synthase has two parts, the upper housing portion and the axle within it. As the axle spins, it comes in contact with the housing, causing an interaction that is thought to synthesize the ATP. However, since it is not possible to see the movement of the ATP synthase directly, it has not been possible to further elucidate this mechanism. To examine the minute movements of the exceedingly small protein with the human eye, attention was drawn to the method of creating simulations using computers. This is a simulation of the movement of the ATP synthase. The calculations were based on such principles as the strength of attraction between the molecules and fundamental laws of physics, such as Newton's equation of motion. Let us focus on the central axle, which appears red. It spins with an irregular motion, knocking into the surrounding housing. In fact, it spins as fast as once in 5 milliseconds, or 5/1000ths of a second. We are seeing what occurs in that instant. This is the image seen from below. This movement of the ATP synthase first became known through computer simulation. Anticipation is growing that in the future, the new supercomputer can be used to perform more detailed simulations that will open up the mobile world of tiny proteins.
今まではライフサイエンスにスーパーコンピューターっていらないよって言われてたんですけど、やっとそのライフサイエンスで、このスーパーコンピューターを使うことで、世界が変わっていくっていうことを周りからも認知されてきた。コンピューターっていうからちょっと分かりにくいんですけど、これある意味そのものすごく高性能な顕微鏡があるようなもんですよ。で今まで持っていた分解能の100倍の分解能の顕微鏡があったら今まで誰も見られなかったものが見えてくるわけですよね。でそれを最初に使って見ると多分見ただけで新しい発見が出ますよね。でさらに見続ければ、今まで気が付かなかったいろんなことがいろんな分野で見えてくるわけですよね。それを期待してます。
The supercomputer will be used to perform simulations that explore the essence of life. And as the completion of the supercomputer approaches, the research of each group in the next generation integrated simulation of living matter R&D project intensifies. The Molecular Scale Research Group is focusing on a problem that has serious social implications. That is multiple drug-resistant bacteria, against which most antibiotics are ineffective. Shoji Takada's group has been pursuing this research in the hope that a simulation might shed light on the mechanisms of certain proteins in drug-resistant bacteria and lead to a solution to this problem. Bacteria die when medication penetrates their cellular walls and takes effect. But drug-resistant bacteria do not die after medication enters them. We know that one of the reasons is that a protein complex called a drug efflux transporter operates in these bacteria cells. The drug efflux transporter is embedded in the cell membrane and is comprised of a number of proteins. It functions by expelling drugs that enter the cell. As a result, the medicine has no effect, and the drug-resistant bacteria survive. It was speculated that the mechanism worked as follows. The part that expels the drugs, referred to as ACRB, is made up of 3 proteins. First, a mouth opens below and takes in the drug, which is then expelled from an opening above. This process is thought to be repeated by the 3 proteins in succession, thereby expelling the drugs. To test this hypothesis, Takada decided to use a simulation to reproduce the action of the drug efflux transporter in expelling the medication. The simulation was performed with the following method. The proteins are made up of a variety of amino acids, but using a technique called coarse-grained modeling, these amino acids are treated as larger granules. This allows for more efficient computations and brings the overall picture into focus.
これは、多剤排出トランスポーターでして、ちょうど、細菌の内部がこの下側になりまして、上側が細菌の外側に当たります。ですからこれが、薬剤でして、トランスポーターが薬剤を結合しているところを横から見たものです。
This is a side view of ACRB, the part of the cell that expels the drugs. This simulation focuses on the central portion. It has a width of 9 nanometers or 9 1/millionths of a millimeter. How does the efflux transporter expel the drug? To make it easier to see the drug, seen here as yellow inside the transporter, the part in front of the drug is transparent in this display. The transporter sways gently because heat is shaking the molecules. Using the energy from the shaking, the drug is slowly pushed upward. The drug finally nears the opening, and it is expelled. Now let's look at this from above. Note the part that will expel the drug. It sways gently until, after a while, the part that expels the drug creates a large opening and the yellow drug becomes visible. And then it is expelled. On a scale of less than 10 nanometers, the event takes place in less than 0.1 seconds. It is a minuscule world that one could never see, but a simulation opens up the possibilities of explaining the mechanisms of life.
分子生物学の教科書を見ると、分子っていうのはこう名前だけ持っていて記号のように見えたりするし、 結晶構造を見ていると分子は止まって見えるわけですけれども、でも実際には分子はこんなふうにフラフラっと動いていて、その動いてフラフラっとしていながら、でも最終的には何か仕事をしていると。タンパク質が動くっていう働くっていうのはそういう揺らぎの中で働くということなんで、それが本当に正しい姿だと。
Based on these results, there is a plan to use the supercomputer to perform more precise simulations in an attempt to reveal the mechanism of the drug efflux transporter. This is the work of Mitsunori Ikeguchi's group in the molecular scale team. Here we will perform a simulation not with the coarse-grained model on the left, but with an all-atom model that faithfully reproduces all of the atoms. Also taken into consideration are the molecules of the cell membrane the ACRB is attached to and the water molecules that surround the transporter. That is this image. Because of the massive number of calculations, this represents only some 100ths of nanoseconds, some 10 millionths of a second, but the condition of the shaking atoms was reproduced. Let us focus on the zone that captures the drugs which are indicated in yellow. This is the area marked by the white circle. The process of gradually creating an opening was reproduced. This becomes the entrance for the drug to be ingested. There is great anticipation that, when calculations on the new supercomputer become possible, the mechanism behind ACRB will be further revealed.
物理科学の方でいうごくオーソドックスな水素結合だとか、そういう基本的な原子の相互作用の原理からですね、こういう大きなタンパク質、この多剤排出であれば薬剤を排出するというようなですね、その生命現象に非常に重要な機能っていうのを明らかにすることができるというところが、非常に厳密計算というのの面白さだというふうに思います。
In the future, if simulations like these fully elucidate the mechanism of the drug efflux transporter, it may be possible to contribute to the solution to this problem by stopping the process of expelling drugs or developing drugs that will not be expelled from the bacteria.
一番その我々の強みといいますのは、科学物理でやっている、論理をそのまま持ち込みますから、そこで見えることは相当程度厳密な正しい結果が出てくるはずなわけです。ですけど今はまだ理解をするっていうレベルにとどまりますけれども、これからいろんなことができるというようになってきますと、予測ができる、それから欲しい機能のタンパク質を作ることができるっていうモデリングができるっていうところまで拡張することができるようになってきます。そうしますと、今までの、科学の中に、限定されていた問題が本当に例えば医療の現場、製薬の問題ですとか、そういうところに使われるようになってきて、一般の方にもその力、計算機の力というのを理解してすぐに肌身に感じて理解していただけるようになるんだというふうに想像しています。
The Integrated Living Matter Simulation R&D project aims to simulate cells and organs based on this type of protein behavior and ultimately to produce a three-dimensional simulation of the human body. To this end, organ and body scale R&D is being carried out. It is now possible to simulate the formation of a blood clot in a blood vessel. This is actual footage of circulating blood flowing from right to left on the screen. The small white particles are platelets sticking to the wall of the blood vessel. When they accumulate, a blood clot forms. This is a close-up of the platelets, the white particles, sticking to the vessel wall. Here we magnify a single platelet. It is 2 micrometers in diameter. Many long, thin, thread-like extensions surround it. Here we magnify these thread-like extensions. The tip of the thread adheres to a protuberance on the wall of the blood vessel. The elements displayed in red and blue are adhesive proteins. They are each just 4 nanometers in size. When the 2 proteins join, the platelet begins to adhere to the wall of the blood vessel. Data on this phenomenon on a minuscule scale was incorporated in a simulation that was performed. The area indicated in purple is an injured part of a blood vessel. The simulation shows how the adhesive proteins make it difficult for the platelets to flow by when they come into contact with the injured area. This leads to the formation of a blood clot. Continuing research in this area, it is hoped, will prove valuable in predicting diseases that result from blood clotting. And the research being pursued in the lab of Toshiaki Hisada and Seiryo Sugiura's of the organ and body scale team is the heart. The heart functions as a pump that sends blood to every corner of the body. This precise simulation of the heart was produced on a currently available supercomputer. It is also possible to simulate the ejection of the blood that flows within the heart. The blood pressure the heart produces and the electrocardiogram can also be depicted. Now at the Hisada lab, a simulated heart is being built by combining each one of the individual cardiac cells. First, the dynamic of these muscle cells was faithfully simulated. Then, using a theory called multi-scale analysis, a countless number of these cardiac muscle cells will be combined to create a more real simulation of the human heart. With this simulation, it will be possible to reproduce diseases caused by changes in the tiny proteins that comprise the cells. For example, vinculin is a type of protein that connects cardiac muscle cells to each other. A lack of vinculin has been suggested as a possible cause of a disease called dilated cardiomyopathy. A simulation was made of this condition. On the right, where vinculin is absent, the adhesion between the cardiac muscle cells is weak, and the white component becomes stretched. As a result, the amount of blood ejected from the heart is greatly reduced. You can also see that the red portion, which indicates a fast blood flow, is smaller on the right side. At the same time, the blood pressure doesn't change much. This is thought to result in the dilated cardiomyopathy condition. In this manner, it is possible to illuminate how changes in a minuscule protein will affect the functioning of the heart. It is hoped that simulations on the new supercomputer of the precise composition of the cardiac muscle cells will be useful in the diagnosis and treatment of heart diseases caused by changes in proteins.
私たちがやってることが、将来の医学のに対して、ある種の変革を与えうると信じてやってますし、やがて本当にそういうシミュレーションがアシストすることによってより早く病気が見つかるとかよりいい治療法が検討されるっていう方向に向かうと信じて日夜やってます。
In the future, through the detailed dynamics of various proteins and organs recreated through precise simulations on the supercomputer, the essence of our life activity may become visible.
今私は生きていますし、皆さん生きて活動していますよね。で、これは生命現象そのものはですね、あまりに複雑で誰も理解していないし、予測もできないし計算不可能な世界ですよね。でも秩序がある。でも今回 K で 私たちが挑もうとしているのはその生命現象の中のある、要素となる現象を動的にその時間的に変化するものとして変化していくその変化することで人が生きているその、一つの要素を取り上げて、動きとして計算しようとしてるんですね。でこれを積み重ねると生命現象は説明できるはずだと。
Let's take a look at the research results of the other teams in the next generation integrated simulation of living matter R&D project. First, the Brain and Neural Systems team. The human brain is comprised of about 100 billion neurons. Thought and memory are produced by forming networks of neurons. This is a neuron in the process of extending itself. When neurons are growing, they add branches and connect to other neurons, forming networks. Movement at the extremity of a neuron has been simulated. Inside the black circle, representing the cell membrane, are many rod-shaped actin filaments. They are 5 to 9 nanometers in length. The actin filaments individually press against the circumference, causing it to move. This is the first step in a future simulation of the growth of neurons in the brain. Further, a simulation has been made of the transmission of signals among neurons that have formed a network. This simulation presents about 70 neurons that have formed a network and are relaying signals. In the future, the supercomputer will be used to simulate larger numbers of neurons in an attempt to approach the function of the brain itself.
脳というのは非常に興味深いシステムであって人体の臓器でありながら我々の全身を制御する中枢コンピューターの役割を果たすわけですね。でその脳の中には100億から1000億個のニューロン神経細胞がありその神経細胞の中には無数の分子があります。脳を理解するためには運動というものが、神経回路によって成り立ち神経回路の中に神経細胞がありそして分子があるのだというこの階層全体をまとめていくことによってしか理解できないというふうに考えておりチャレンジングなプロジェクトだと思っております。
Next, let's look at cell-scale research. This is a cell simulated in a computer. The blue portion is the nucleus, and the green components are mitochondria. Precise reproduction of cells, including their internal components, has enabled the simulation of the material exchanges within and between cells. This is the transmission of a calcium ion within a cell. Calcium ions function to activate various components of a cell. This is a simulation of a cell responding to the stimulus of another cell. The exterior stimulus, indicated in green, reaches the white capsule called a dense granule that stores matter, and it then turns red. Then matter, indicated in blue, is emitted from the cell, passing the stimulus on to other cells. This may provide clues to understanding the exchange of matter between cells, which has long been a mystery.
細胞スケールのシミュレーターが出来上がった際には細胞だけではなくて組織の状態での細胞の振る舞いの、計算ができるようになると思います。最終的には、患者さんのですね体の中の、疾患の時 疾患時の細胞の振る舞いですね、こういった解析ができるようになると思いますので、その治療方法であるとか薬剤の開発の役に立つシミュレーターになり得るんではないかというふうに思っています。
Next is the Data Analysis Fusion team. The human body is said to contain tens of thousands of different proteins. In most cases, those proteins perform their functions by working with other proteins. This group is using an entirely new methodology to illuminate which proteins work in combination in an attempt to solve the riddle of life activity. Let's take a look at this methodology. An assessment is made as to whether two proteins will work together by analyzing the uneven shapes of their surfaces. This map displays the results of this assessment. For example, the white and green proteins will work together, so the map displays a red mark, while the white and blue proteins will not work together, and the mark is green. Using the new supercomputer, it will be possible, for example, to analyze the combinations of the hundreds of proteins involved in cancer, and perhaps to shed new light on the mechanism behind the disease.
生命を演劇に例えると、タンパク質はその舞台に登場する役者です。私たちはその舞台にどの役者が何人登場し、いつ舞台から裾に消えていくか、それを演出している生命のプログラムをデータ解析という手法であぶり出して、そして動的に変化していくその生命のプログラムを捉えようとしています。抗がん剤が効く人と効かない人がいます。その抗がん剤が効く人の生命プログラムはどのようになっているのか、効かない人は生命プログラムがどのようになっているのか、そういう違いを明らかにして、では私の生命プログラムに対してどの抗がん剤をどういう順番で適用すればより良い治療を受けられるのか、そういう応用が開けてくると思います。
Finally, the High Performance Computing Group. In preparation for the full powering up of the supercomputer, the group is working on developing methods to maximize the exercise of its computing power. These new methods are necessary because the number of CPUs in the supercomputer is on a different level of magnitude. There will be 80,000 CPUs, while computer cores will number 640,000. To maximize computing power, the CPUs must exchange data in the most efficient way possible. In their basic research, the team focused on the pattern of communication between the CPUs. Until now, information was exchanged between the appropriate CPUs whenever it was necessary. However, with this method, serious problems occur when the number of CPUs multiplies. When data accumulates at one spot, a traffic jam in the flow of data occurs. In one of the team's proposed patterns, data is divided and communicated in 3 directions on the X, Y, and Z axes. At first glance, it appears to be circuitous, but the exchange of data occurs systematically, and traffic jams are less likely to occur, even with many CPUs. By further exploring the issue along these lines, the group is searching for the best possible communication pattern for the computer.
計測コンピューター K の力を 100% 引き出して、それの上でのシミュレーションをより早く動かすによって科学的な成果をたくさん引き出してくるというのが、まず一つ重要なところです。将来は100万あるいは1000万あるいは1億といったようなコアを並列に動かさないと計算機の性能を引き出せないという事態になってくると思われます。でそのための一つの準備として、我々の開発みたいなのが長期的に生きてくるのではないかというふうに考えています。
Using the supercomputer to explore the essence of life through integrated simulations is an R&D project that is unprecedented in the world. Koji Kaya, the director of this project, believes that it will mark the dawn of a new age in simulations.
こういう生命科学のシミュレーションが我々個人の生命の問題にまで立ち得ると、そういう時代になったという時に、研究者の在り方そのものもやはり生きていることということをもっと深く学びながら、そして社会の方々と語り合いながらやっていくべきだという問題提起をしたとことだと私は思っておりますし、いかにコンピューテーションが重要であるか、また皆さんにとってもどのような影響があるかということをお考えいただければありがたいと思っております。
Construction continues with the target of having the supercomputer up and running in 2012. Then, the next generation integrated simulation of living matter R&D project will finally launch. New worlds are about to be opened in the fields of medicine and pharmacology, while the effort to answer the fundamental question, what is life, will continue.